Pennsylvania State University College of Medicine in Hershey. And we are ready for listening to your talk. So, Don, it's a real pleasure to give you the virtual floor.
Well, thank you, Jan. Thank you, Martin. It's really an honor to do this. I'm really very happy that you thought of the idea. Really interesting times right now, and most important thing is that we continue on. There are all kinds of obstacles in science, but right now, but we are doing things like this and overcoming them, and things will get better soon enough, and or maybe not soon enough, but they will get they will get better. Okay, you can hear me okay, right? And I can't see or anything because I have the full screen up, but you can hear me and see me?
It's perfect, Don, perfect.
Okay, all right. So I'll go to the actual presentation. So I don't need to do much of an introduction. As you know, I'm at Penn State College of Medicine in the physiology department. Motribac's in the same department. We have a lot of good people here who work on a lot of signaling mechanisms. Calcium signaling is not the only thing we do in the departments, for sure. I thought that the first thing I would do is just say a few words about Mike. There have been many words said, and but I think that everybody was very shocked by Mike's death just this year. Such a strange time since then, but Mike, I visited many times, and in the mill, I put these pictures on the web, but in case you didn't see them before, that's where he used to live. Just the most beautiful place in Suffolk, not far from Cambridge, and he was very happy there for many years, and just enjoyed the gardens. And I just like that picture. You see the look on his face. He's sublimely happy. And that's Sue, his wife, and there is some noise. I was talking of. Somebody might not be muted. Okay. So the funeral was a very great celebration of Mike's life. Some tears, but a lot of sort of just thoughts, happy thoughts about how good Mike was, what great life he led. And these are some of the pictures from Trinity. It was a beautiful day not very long ago, and that's actually Robin standing in front of Mike's office, his old office in Trinity, and kind of interesting. But it's kind of amazing how since that time, so that was the 2nd of March, and it's like the world has turned on its head since then. That was just before things started to close down in a serious way, and so travel and all kinds of things are now stopped. Our labs have been closed for a long time, but I think that we're all seeing this light at the end of the tunnel, and we're pretty convinced it's not a train coming towards us. That it's just a question of, it's a long time, and each country has its own tunnel, and it's taking its own time. But things are getting back slowly, and you know, within a few months, things will be a lot better, and we'll just take it like one day at a time. So today, these were sort of words from Mike. You know, he always said, from conception to death, calcium signals control a vast number of functions, and of course they do. And I don't think there's anybody listening in who doesn't already know all the things that calcium does in cells. And these events in cells are all controlled by a whole variety of different channels. And I'm not going to go through all of these. You can see my pointer, okay? Yep. Is that right? You can see the pointer?
Yeah, yes, we see the pointer. No problem.
Okay. Sorry, it's not very big. I could. Okay, so lots of different channels get activated in very different time scales, or from microseconds up into seconds, and many different events being activated. And the channels that we've worked on particularly are the very slow activating channels, and they control slower events like metabolism and transcription and cell fate. So, so as you all know, for years we have been studying the STIM and Orai proteins after the pioneering work from a number of labs, Anjana, Rich Lewis, Mike Cahalan, ourselves, and a great number of people who contributed to the identification of STIM and Orai. The STIM proteins are all over the ER, and the Orai channels are distributed all over the plasma membrane, but they're in very different membranes, and they come together into junctions which we never knew about until we could see STIM and Orai, and now we can see them very well, these little junctions between the ER and the plasma membrane, and the STIM protein aggregates there and actually binds to the plasma membrane. In fact, we have another, and you've seen this several times before over the years. It's pretty much still the model, few changes, but the STIM protein is in the endoplasmic reticulum. This is the junctional space. So this is these close junctions between ER and plasma membrane, where STIM proteins exist in the ER membrane always as dimers, and they when the luminal calcium decreases, then the calcium dissociates from the STIM EF hand sites inside the lumen of the ER, and this triggers an event that slams the two N termini together and causes the whole of the C-terminal region here in the extracellular, in the junctional space, to undergo a big conformational change, and it becomes elongated, and it's able to actually interact with the plasma membrane. So it will move into these preformed junctions, and there these polylysine residues will interact with acidic phospholipids and bind pretty tightly to the membrane and cause what we call, and we will see these puncta of STIM proteins in the between the endoplasmic reticulum and the plasma membrane. And then in that state, when it's bound and it's associated with the plasma membrane, it's able to tether Orai channels, which are these hexamers in the cell membrane, and they get tethered and they become activated as a result of an interaction between this the STIM protein and the Orai channel. So, you know, a few years ago, this junction sort of started to become really complex, and there were all kinds of proteins which were sort of viewed as being important for controlling the what we call this calcium signaling junction. So many of these proteins, like SRAF, CRACR2, post, these are interesting proteins, and Golly was another one, but we don't hear quite so much about some of these, at least, and so they're there, but we're not necessarily sure that they're essential for the STIM and Orai interaction, which is actually quite a simple process in a way, and STIM and Orai can work very effectively to interface with each other and activate calcium entry without other proteins there. And I think that was shown by Patrick a number of years ago when he put reconstituted STIM and Orai in yeast membranes. So so these two proteins are sufficient to cause the calcium entry channels and calcium entry. So our question has really been, how does the STIM protein actually dock and interact with and activate the Orai channel? And there's a lot of other people asking that same question. But if you look at the antibody, the STIM protein looks a little bit like an antibody molecule. So it has two sites on it. There's a dimer, and so we're going to talk about these binding sites. This is the very important region of the STIM protein called the SOR, or STIM Orai activating region, or the CAD region, as named by Rich Lewis. This is the name that Shmuel Mullen gave it when he discovered this in a beautiful paper, originally Nature Cell Biology, and then Rich Lewis's paper came out shortly after, immediately after, in Cell, and they were able to show how important this STIM Orai activating region is, and it remains sort of occluded in the folded up STIM protein, and then when STIM gets activated, it gets exposed. is then able to interact with the plasma membrane or with the Orai channel and tether and activate the Orai channel. So the thinking had been that the channel, although only 2 subunits is shown here, it is a hexamer, and we've established that. Many other people have established. Steve Long, from his crystal structure, showed that this really does operate as a hexamer. I'm just showing 2 subunits here, sort of across the channel, forming the channel, but there are 6, of course. And the thinking had been that the dimer of STIM, or dimer of SOR, as it interacts with the Orai channel, is actually interacting with across 2 adjacent Orai subunits. And the crystal structure for this came out from Schoen's lab, and for this SOR dimer, there's a very beautiful structure that Shengyu Tsai, who then worked in my lab, showed the structure of the SOR unit, and a very tightly held together, pretty stable crystal structure. And we noticed were certain residues at what we call the apex of this molecule. The F394 residue was one particularly, and we showed, this is now sort of some while ago, that residue was critically important. If you mutated it to a leucine, actually that made it look more like the SOR from STIM2, which doesn't bind so effectively. And this is calcium activation with STIM proteins mutated at this point. And with the L in that point, you get much less activation. Actually, when we mutated it finally to a histidine, then we found there was no activation at all. So this F394H mutation completely blocked the function. And we went on to use that observation, and we made concatenamers of the SOR protein, and we could link together either one that had a histidine there or one that was wild type, and combinations of those. And we were amazed because we thought that both of them had to be able to bind to the Orai channel, but actually we got full activation when there was just one histidine on the one side or one histidine on the other. But when they were both histidines, both of those F394Hs were mutated, then we had no function at all. And that led us to think about the SOR protein not interacting between 2 adjacent subunits, but actually interacting with just one subunit. And that was sufficient for the activation of channel. And of course, going back, sorry, of course, the other unit, if it has a phenylalanine, is able to then interact with other Orai channels and cause them to cluster. So we spent a lot of time thinking about these clusters and looking to see their existence, and we published quite a lot on this. And essentially, we sort of see a lattice. We believe there's a lattice of Orai channels held together by STIM proteins. So, and this was kind of what we call the unimolecular coupling model. So only a single unit from a SOR has to interact with a single unit from the Orai channel. But because these things can vary in size, so the stoichiometry of this can be really quite variable. So it can go, if you have an excess of the SOR dimer, which is shown here, then you can have a 1-to-1 ratio and a 1-to-2 ratio. And when it becomes a large lattice, it becomes a 1-to-1 ratio. So that explained the kind of variable stoichiometry that most people had observed. And we indeed could see those little clusters of the Orai channels holding the being held together by the SOR dimer. And this is a picture that we published a couple of years ago, super resolution imaging, showing that just using SOR here, the SOR dimer with YP attached to it, that if you had one phenylalanine change to one histidine here, now you don't see any clustering at all, but you do see clustering very well when these are both available for interacting with Orai channel. So we thought this was a very powerful experiment because a single point mutation on that SOR dimer was sufficient to completely change the distribution of the STIM Orai, SOR Orai interaction. That was work done by Yandong and Robert. Robert now is at Stanford, and he left a few months ago. So we were very sad to see him go. But this was just a little summary of the ER plasma membrane junction being the clustering of the significance of clustering of channels. So this is sort of showing you that Orai channels can be clustered in these junctions, and we think that probably circuit pumps and IP3 receptors, since Colin Taylor has shown that, and others seem to be quite close at hand around, but not in the junctions. So the clustering of Orai channels is a powerful means of modulating the signals that are coming in through the junctions, and that interaction controls the store refilling and hence the generation and frequency of the calcium oscillations. And so the proximity of IP3 receptors and the pumps to these clusters is probably critical to calcium signal generation and will determine downstream calcium-dependent activation of an FAT. And the cluster size will be controlled by the relative expression of STIM1 and STIM2, and also this negative splice variant, which is able to bind but not activate the channels. And so when this gets into, when STIM2 or even more important, the STIM2.1 splice variant gets in, it breaks down this clustering, and it disorganizes the clustering, and then you end up having a much smaller entry signal, and oscillations are not sustained. So we published and described that in quite some detail. Let me go on to talk a little bit about the interaction between the Orai channel, the hexamer, and the dimer of SOR, which is the active site of STIM, and how STIM actually docks with the Orai channel, and what are the critical interactions involved in that. And just to go a little bit further with the STIM, this is the apex of the SOR molecule, which is the active site of STIM. It turns out that there are two pairs of phi-his pairs, we call them, phenylalanine and histidine, very closely together, oriented quite close together. They both have very important roles in interacting with the Orai channel, particularly this, what we call the outer his pair, which is this pair here, and this is looking at it from the top down here. And when we mutate those things, so this is just with wild type STIM1, where this is phenylalanine and this is histidine. So you have the normal calcium entry, which is activated, and this is fret between the STIM and the Orai. So this is calcium entry after emptying stores. This is the fret between the two molecules. They fret very well, and this is the overlay of the interaction between STIM and Orai. So this is wild type, and when you mutate that, I've already told you, this F394H here, you lose any activation of calcium entry, although APB always brings this back, and it still confounds us, but it's interesting. And there is no fret. Very much, not very, and APB actually will bring back that fret. So that's, I'm not going to talk any more about APB, but this is always a very extraordinary and interesting finding. But when we mutate the histidine at 398, we see something even more interesting, and that is that the activation of calcium entry is completely blocked, and actually the APB effect has gone as well, and yet you still have perfectly good fret between the STIM and the Orai, between the SOR and the actually this is all in full length, between STIM YP and Orai CFP. And if you actually mutate them both, this mutation is dominant. So you actually have effect. So this H398F mutation is dominant over the phenylalanine 394H mutation. So so we find that extremely interesting, because this single point mutation then allows the coupling to happen very well, but there's no activation of calcium entry. And so this is just a little summary. The outer his pair are obviously critical to both tethering and gating of Orai. The H mutation in the F394 prevents tethering and gating. The F398 F mutation, H398F, which will end up giving a phenylalanine pair, allows full Orai tethering but completely prevents channel gating. So so the H398 residue is critical to as a critical determinant of STIM Orai coupling to induce the Orai channel gating. So we really believe that that interaction with this Orai channel is sufficient to cause the activation, and just changing this, you've lost that activation completely without losing the binding. I'm going to switch over to talking a little bit about Orai channel. So this is kind of like the active site of STIM. And checking on time, we got about 5. We did start a little bit afterwards, so I might go on for 5 minutes. Is that going to be okay for another 10 minutes that we talk? Is that okay?
No problem at all.
Okay. If you get bored, you can always turn off. So so this is the Orai channel from the Crystal Structure by Steve Long, Drosophila Orai. And we noticed, and everybody has noticed, that there's six-fold symmetry around everywhere, except on the outside. These two, what we call the M4X peptides, these are the extensions of the last transmembrane domain. They stick out and they interact in an anti-parallel way. So one of them is straight here, and one of them is completely bent, and they're bent over, and they form between those leucines 273, 276 in Orai 1, which are equivalent to the residues in Drosophila. They cause this thing to interact in a way where we began to think that this was perhaps making a binding pocket, and that the interaction of STIM with these, we know these are really important for the interaction of STIM, and we thought that this conformational changing the hinge flexion, and then the coupling to the M3 causes eventually small movement in the M1, the pore forming domain, pore forming helix, and which causes opening of the channel. And so the significance of these are the M4, what we call the M4X peptides here. This is the bent one, this is the straight one, and they're being held together by these interactions between these two pairs of leucines. So we see those in all of the Orai channels. This is Drosophila Orai, Orai 1, Orai 2, Orai 3. They might be isoleucines sometimes, but they're either isoleucines or leucines. So we asked what the significance is of the interaction between these pairs of leucines in the structure of Orai 1. And we asked, why did the two adjacent M4 peptides form an antiparallel complex? Do the zippered leucines form a binding pocket within which STIM binds, as I just described? And we asked also if the leucines have to remain zippered for STIM binding to take place, or must the M4X peptides dissociate, those leucines get broken in order to allow STIM binding? And indeed, we asked, does the M4X peptide bind independently of STIM? And so a big clue to these questions came from when we looked at the D-Orai crystal structure more closely that Steve Long had made, and we realized that the M4 peptides are kind of held together in this threefold symmetry. So this is the crystal structure, which Steve Long, a very beautiful structure. Everything about this is completely correct, but we do wonder whether this zinc, which was used in the crystallization and interacts with histidines, because going back, there's these histidines at the end of the M4X. There's a pair of them here. There's actually 3 of them, but this pair seem to interact with the zinc, and they force those in this crystal structure. The threefold symmetry seems to be forced by the zinc, and so and that allows these leucines in each of those pairs. Here are the leucines interacting with each other. So this is all part of the structure, and it made us think that these interactions between the M4X proteins, peptides, were important for STIM binding. So we decided to take the peptide and to try to see, we knew this is very active site for the activation, for the interaction with STIM. So we took the peptide, the complete peptide here, and these are the peptides from either Orai 1, 2, or 3. Orai 1 is about 30 amino acids. Orai 3 is only about 20 amino acids. And they all start off with a similar sequence, and then they change. So we took that, and we took the M4X peptide, we attached it to CFP, and we put it on to express it with Lin kinase and this construct that we got from Korzeniowski and Cornell, and so to make it only in the plasma membrane. So it's like a little facsimile of Orai, that you take the M4X peptide and you attach it to this construct and have it expressed on the plasma membrane. It worked extremely well, and actually we could see when we co-expressed this construct, this link kinase construct with SOR dimer, which we've used many times. We could see that actually just when they put the link kinase without the M4X peptide, we didn't see any binding of the SOR dimer. When we took the Orai-1 peptide, we saw some binding of the SOR peptide. When we took the M4X from STIM from Orai-2, we found much better when you could see this binding of the SOR now very effectively, and even better still with the peptide from Orai-3. So these are all not very quantitative, not very accurate, but at least showing some indication that the peptides do bind, and the Orai-3 one was better than the Orai-1. And so we started to look and quantitate FRET between these, which is a very good measure for the interaction of those two molecules. So FRET between the CFP M4X construct and the SOR dimer. And we could see FRET. Look, this is with the Orai, the M4X from Orai-1. See a little bit of FRET. And when we mutate the 273, we saw it decrease. When we mutate both of them, we saw the FRET decrease even more. But the FRET was kind of low. And when we went to Orai-2, we saw a little bit higher FRET, and again, mutating one of the leucines, in this case, the isoleucine, you lowered that FRET. But the most impressive thing was using Orai-3. So this is the Orai-3 sequence here. It's much shorter. And when we attached that into our construct, we found very large, very strong FRET. And when we mutate the L282, which is the equivalent to the L273, oops, sorry. We showed the FRET was decreased. This is just binding without the peptide alone. This is very impressive now. So now when you mutate, this is just looking now at the Orai-3 peptide, which binds extremely well to SOR. When you mutate the L282, you see much decreased FRET. When you mutate the L285 to D, you see almost no FRET at all. And then when you mutate both of these, you see also very little FRET. Almost the same as the control where you don't have the peptide at all. Very impressive, strong interaction. And that was what was even more impressive is that the interaction of that peptide with SOR was pretty much the same as the interaction of the Orai channel with SOR with those same mutations. So this is the 282D and this is the 285D, leucine modification. So this one much more powerful than the other one. So it looked very much similar, and the combination reduced it to almost nothing. So we began to think, okay, so maybe these peptides bind together to form this, the leucines actually interact with each other, and that's forming the binding pocket. Maybe that's how it interacts with the SOR protein. So we did everything we possibly could to look at FRET between these peptides. So we can't necessarily rule out there's no interaction, but we could not see any change in the interaction of these peptides when we mutated the leucines on either the Orai-1 or the Orai-3 peptide. So Orai-3 didn't interact. Orai-1 didn't interact with Orai-3. Mutations didn't. Everything we did, we could never see any significant FRET between these things, which makes us think that the peptides are not interacting with each other in order to interact with the SOR protein. And in fact, with the Orai-3 peptide, there are 4. This is a perfect amphipathic helix with the leucines spread out every 3 or 4, and so we could actually mutate all of those any of those leucines, and we saw big effects. So this one gives a medium effect. This one, which I already shown, 285, causes a big effect. But even the other 2 are also pretty large effects on decreasing the interaction between the peptide and SOR. So pretty impressive. And if we mutate those, so we know if we make the L285, the most effective one, into a D, we see no FRET. If we make it into other hydrophobic residues like phenylalanine, methionine, and tyrosine, so we see much decreased FRET. So it means that it seems like the leucine is really important. A small hydrophobic unit is that a larger hydrophobic one won't really do, that it will actually lose its ability to bind. So that tells us that these things are probably fitting into a tight interaction with the SOR molecule. And so we went on and we showed that the interaction between the peptide and whole STIM was exactly the same. This is a time course of FRET interaction, and these are the mutations in the whole STIM molecule. And you can see this gradation, the 285 more effective, 282, and the combination of the 2 not much more effective. And then we ended up by looking at FRET between the whole Orai channel on the whole STIM protein, and exactly the same pattern. So when you mutate 1, 282, the Orai3, you have a very decreased fret, but when you mutate both of them or one of them, the 285, then you have, so the pattern was exactly the same. That means that the peptides are really faithfully emulating the function of the interaction between STIM and Orai. So then we went to function, which is the most important thing. So to see now, if we did the same mutations in function, how do they change the activation of the channel? And so this is emptying of stores, and with Führer just measuring calcium going in. When we mutate, you can see how powerful the effects are. Even the 282D has a pretty powerful effect, but not as strong as the 285D. So that same pattern is repeated here as well. Actually, this is the wrong slide. This is the slide I should be showing. So, so this is the Orai3 interaction, and this is the L285 mutation is almost completely knocks it out, and the combination of the two causes an complete knockout as well. So the specificity of the interaction exactly mimicked by the interaction of the peptide by itself. So this is interesting because Orai3 is also activated by APB directly, slowly but directly, and you can see that these mutations have just no effect, and they don't alter that at all. So these mutations are purely to get activated by STIM. They have no effect on the activation by APB. So there's no specificity there at all. This is just the same with the electrophysiology. So and interestingly, you can use those peptides. This is the last slide. Last data slide. That the Orai3, so this peptide, when we express that construct with the Orai3 M4X, it will, this is just native store operated calcium entry in a hex cell. And when you just express the O3 MX4 peptide, it really blocks almost all of that. And if you mutate the L285, it doesn't block at all. And this is kind of interesting because the Orai3 peptide really blocks very well. The Orai1 peptide is much lower affinity for the interaction with STIM, and so it gives a much smaller effect. All right, so these are just conclusions, sorry, a little bit long, but the binding of these peptides exactly recapitulates the binding of STIM and SOR to the Orai channels. So the M4X peptides are the critical binding sites for STIM proteins. We see no interdimer fret pairing of those M4X peptides. So the critical leucine residues are not involved in direct interactions between the M4X peptides. Indeed, those leucines are the sites for STIM interactions. So our findings that each M4X helix peptide independently interacts independently with STIM1 strongly supports the recent structural evidence that closed Orai channels can exist with unfolded, unpaired M4 helices. So that pairing does not seem important because new structures coming out now seem to show that they can, the channel can exist without those things paired, and it's not necessarily open. So STIM-Orai coupling is mediated by the interaction of a discrete site on the apex of the SOR domain of STIM proteins with a small discrete C-terminal peptide of the Orai channels. And so this mechanistic paradigm is, of course, very consistent with the unimolecular coupling model, this small direct interaction. And just to end up, I'm not expecting you to follow this, but in the new structures coming from Steve Long and Shen's lab in Tianjin, in China, the first cryo structures again show no pairing of these between the M4X extensions, and indeed Steve sees crystal structures where these things are completely unlatched, and yet the channel is not necessarily open. So the unlatching of these doesn't necessarily open the channel, but it certainly seems to be required for the interaction of STIM with the molecule to activate it. So I'll end that. We can go back to some of these models if people have questions, and then just thank the lab. These are members, particularly Robert, who's now left, as I said. James did a lot of the work with the peptide. Yandong, who's been in the lab, of course, for many years, and then Lane and Juliana and Sarah, who's just starting now, are important members of our lab as well. Here we are all sort of huddled looking at structures, which we do every day. And also should thank Eugene Wang's lab, used to be in my lab, is very effective now in China, in Beijing, and also Chubak lab, who we interact with constantly. So we thank many people who we've interacted with over the years, including, of course, Mike Berridge, and to our funding. And so I'll stop there, and we can leave some conclusions up, if you like, and I'd be very happy to take questions. So I assume there's now some virtual applause if there's anybody still awake or still online.
Thank you very much, Don. It was really impressive work. This was really impressive work and a very good talk. So we were with 135 people listening to you, 135 people from everywhere in the world. So I think it's a big success. The talk is now open for discussion, but as Martin asked in the beginning, we expect questions to be introduced by chatting, because otherwise all the microphones will go on and nobody will hear anything. So if you have a question, please fill it in the chat box and I will relay it to Don. And while you are doing so, I will ask the first question myself. Don, it was very interesting to hear how STIM and Orai interact, but in the beginning you had also that slide with all those other proteins which were part of the complex, or were in the neighborhood at least of STIM and Orai. Can you tell a little bit more about which are the important proteins there and what is their function there?
So I would say overall we had been a little disappointed with looking at each of the different proteins that have been published, and not a whole lot is still being published on this. But these proteins are several of them are important for modifying the interactions of membranes, modifying the ability of membranes to come close together. And so it would make a lot of sense that there are proteins, that those proteins and probably many others control how large these junctions become. They control perhaps whether they can even form directly. So my feeling is that those proteins are generally altering the domains of the junctional domains, as opposed to being direct modifiers of the STIM-Orai interaction. Now, I may be wrong on this. There's been a lot of work on Seraph, which does look like it's a very interesting sort of modifier of the function of STIM. It's also in the ER membrane. So I would say that the questions are still open on many of these proteins. I have the suspicion they don't necessarily modify the direct interaction of STIM-Orai or bind directly to the interface between STIM and Orai, but they do seem to control the formation and the ability of junctions to allow STIM and Orai to come in. But you know, the point I made at the beginning is that STIM and Orai can work very effectively by themselves. They don't need other proteins to interact, but there's no question that junctions like this must be controlled by a lot of other proteins.
Okay, that's very clear. Thank you. We have meanwhile a question from Geert Bulting from Leuven, Belgium, and he's asking whether there is a physiological role for zinc in the STIM-Orai activation process in living cells.
Wow, that's a really good question. It's like, we would love that to be the case. We're not sure. And I have to say, we haven't really talked to Steve Long. I'm not sure if he's on the line, about that zinc observation. But we were kind of perplexed at the, I could go back to the picture. Did I already go past it? I think I did. Yes, it's just in here. Zinc. So whether zinc might be binding in there, I don't know. Whether there's enough zinc in the cell to do what they found with the zinc there. I don't actually remember now what the level of zinc was in the crystals, but those two histidines, right at the end, are this pair of histidines are very powerfully. This might have been really the key to having the crystal structure form. I don't know. Lots of things happen in crystals, but everything about the structure is all sort of makes complete sense. The six-fold symmetry, but this threefold symmetry could have been forced by the zinc. And the zinc would therefore have just held the histidines in place. And then, so these are the histidines are sort of at the end here. Only one set of histidines, not on the other end of the TM4 are involved in that. But those would be held together, and that would force these leucines to interact with each other. So then when people found that those leucine mutations were really important for changing STIM and Orai interactions, they began to assume that those were necessary for STIM to interact with Orai, and they are, but they are there for not to create this sort of binding pocket, but are there to allow the STIM to actually interact with Orai. So it's possible in the resting state that these leucines really do interact in this sort of zipped up fashion, and maybe that's what keeps the channel generally in the closed state. So we just don't know whether this structure really exists in the native Orai channels, and this is an important question. So it is a great question, and it's just possible that zinc in the cell might indeed keep in that state.
Okay, then we have a question from Anthony Morgan from Oxford University, and he's asking whether you have an idea about how ML9 might be inhibiting in your model.
ML9. Forgive me. What is ML9? Did you say ML9?
ML9. I know it's an inhibitor of something, but
right. I recall that ML9 was something that modified STIM. I'm sorry. In these sort of situations, it's not easy just to ask the question. I'm sorry. What was the ML9 effect? I remember Jim and others published a lot on ML9, but now I've forgotten the effects of ML9. Perhaps that
maybe Anthony can in the chat box give a little bit more information about ML9, and meanwhile we will take the question from Catherine Leclerc from the University of Toulouse. And she is asking, what about the interaction, and that is something I do not know myself, between LTTC and Orai STIM. I do not know what LTTC is as a protein. I hope you know. Otherwise, Catherine has also to clarify this.
Are you talking about maybe the L-type calcium channel? The is that what we're because it was actually in that picture of we published, and so did Roger Chen, on L-type calcium channels interacting with STIM, and in fact being inhibited by the STIM proteins, and quite a few people have followed up on that, and the observation is quite clear. And so we showed that the L-type calcium channels were also in those junctions when we overexpress them, at least. So, and we had the idea that perhaps STIM, while it activates Orai channel, it inactivates L-type calcium channels. And we had all kinds of models based on the idea that perhaps during remodeling of a smooth muscle after injury, in the injury response, that perhaps STIM was important for now not only activating Orai channels, which is important for the proliferative events, but also might have been important for knocking down the function of L-type calcium channels, which are generally there in the contractile response. So we haven't done very much more on that, but it was certainly a very interesting possibility. I hope that's what was the question meant, was LTCC?
Yeah, yes, it was that indeed, yes.
Yes. Okay.
And meanwhile, Anthony Morgan clarified that ML9 is an inhibitor of STIM1 oligomerization. It's also a kinase inhibitor. But it inhibits the oligomerization of STIM1.
Spent quite a lot of time with ML9, showing that it could prevent the aggregation and the punctual formation of STIM. So we have not looked at that for, I think we looked at it a few years ago, but I have to talk to Mo more about that, or maybe Mo has a comment about the ML9 and its effects. So, okay.
Okay, we have a lot of questions. I will take two more questions. I will select two of the questions which are sent in, and then I think we will have to close because time is running. The first question is from Declan Manning, and he's asking about whether you have observations or ideas about STIM and TRIP channels interactions.
Oh, okay. So the TRIP channels, we and many others worked on TRIP channels, and many people are still working on TRIP C channels, particularly because Craig Montel was the one who first showed that the canonical TRIP population channels, which look more like the Drosophila TRIP channel, might be the mediator of store-operated calcium entry. And I think Mike Berridge was very excited. We were all very excited about TRIP channels, and there's still a lot of people working on some very interesting aspects of TRIP channels. But we find that since the Orai channels were discovered, that we have mostly worked on Orai channels, and we have not really gone back to look at TRIP channels. But it seems that there were a lot of papers suggesting that showing that the STIM proteins could interact with TRIP channels. And I think, again, most of those papers are probably referring to the fact that when STIM is overexpressed in large amounts and you empty stores, you change a lot in the membrane. And the TRIP channels are extremely interesting, and they do seem to be quite closely associated with Orai channels in maybe the same domains within membranes. And so it seems that TRIP channels do get carried into junctions, or they may be close to the junctions. But in terms of a specific mechanism of interaction between STIM and TRIP channels versus STIM and Orai, that interaction of STIM and Orai is becoming so well defined, it's so specific, that as far as we can tell, there is no specific interaction between the STIM proteins and TRIP C channels that is on anything like the same lines or maybe the same significance as the Orai channel. So we tend to think that the TRIP C channels are an extremely interesting group of channels which are associated in the membrane and maybe close to the junctions and maybe even in junctions. And they may be having important modifying effects on the calcium entry or mediating calcium entry, but they are not what we call store-operated calcium channels. We do believe that they're not STIM activated. They may be STIM, they may be altered by the action of STIM, but they are not directly interacting with the STIM proteins per se. That's our belief.
Okay, and now I will take the last question. It's from Marek Michalak from University of Alberta in Edmonton, Canada. And he is asking whether you have thoughts about the molecular mechanism responsible for Orai STIM dissociation.
Oh, okay, good. So yeah, it's good that this little slide is up. And I think you can still see the slide. So this process of activation is, very clear, but we frequently don't talk very much about deactivation. But the most amazing part of the STIM-Orai interaction is that as soon as the calcium inside the ER goes up a little bit, these N-termini dissociate with each other, and immediately the whole of this process is reversed. So it remains activated only when the calcium levels inside the ER are decreased. And so that reactivation, the dissociation and deactivation of the Orai channel is a fascinating and crucial part of the whole of store-operated calcium entry. And I'm sorry, I really didn't say anything about that. And we don't work on that necessarily per se. A lot of other people do. And the deactivation by calcium is pretty well understood. Calcium coming out deactivates the channel in different ways. But how the STIM protein actually dissociates and immediately goes back out of the junctions, that has not been studied so much, but it is a very important fact that this is a reversible process. These junctions form and they tickle the STIM protein. The STIM protein tickles the Orai channel to open it for just a brief period of time, and then when the calcium goes back, they dissociate and go away. So it's a kind of a homeostatic process. When the calcium goes down a little bit inside the ER lumen, you get a little bit of interaction with Orai channels. That calcium coming in is very important for activating transcription and other effects, but it remains activated only for a short period of time, and as soon as the calcium comes up, so these things go down. When calcium is depleted in stores, we always talk about depletion when it's emptied from stores, and the whole cell is thrown into oblivion. And so the levels of calcium that change in the ER lumen are really quite subtle, and the numbers of STIM proteins interacting with Orai channels is really quite subtle as well. And we imagine that this reversible process is extremely important, so that you only need calcium, you only have calcium coming in when it's needed. So it's sort of just a lot of words, you know, but the dissociation, yes, is very important, and it is complete, and it's you know, just as spontaneous as the activation, and dependent on the level of calcium in the ER lumen. Good. It was a good question.
Okay. Before passing the microphone back to Martin, who will close, I want to thank you again, Don, for this fantastic talk. It was really interesting. We had a really nice discussion. I want to thank all the people who participated for this, and I'm very happy so many of you could join us. And so thank you, everybody. And Martin, maybe you can say some more words about the future.
Certainly. Can you hear me okay, Jan?
Yep.
Thank you. Okay, well, listen, thanks very much for coming along, everybody. I hope you enjoyed this first webinar. We are intending to continue the series for as long as people are interested in it. So we'd be grateful for your feedback if you'd like to send some in, what we can improve, what you might like to see. We will shuffle the times around a little bit so that we can accommodate different parts of the world more comfortably, because it's the middle of the night, unfortunately, in some parts of the world, and we would like to make sure this is inclusive. We'll have another presentation in 2 weeks' time at the same time, and it will be Sandip Patel talking about signaling through acidic calcium stores. And we will send a link to that meeting through normal ECS communication channels, and we hope you can join us then. So thanks very much for attending, and stay safe, everybody, and hopefully see you in 2 weeks' time for another get together.
Thank you, Martin. Thank you, Jan. I really enjoyed it. I'm very glad this worked. I'm happy to be the guinea pig. And
it's very good.
Guinea pigs prosper.